Polymerase chain reaction, or PCR, is a powerful tool for amplifying a stretch of DNA. From as little as a single piece of target DNA, you can make enough copies for sequencing, cloning, or gel electrophoresis. This exploration will guide you through a PCR amplification. A PCR reaction is typically carried out in a small tube placed in a machine called a thermocycler. The thermocycler simply changes the temperature to selected values for defined periods of time according to a program which you can repeat over and over again. The initial reaction mixture contains a target DNA, synthetic oligonucleotides corresponding to two sequences in the target, DNA polymerase, and a mixture of the 4 deoxyribonucleoside triphosphates. Each cycle consists of 3 steps: denaturation, annealing, and extension. In the denaturation step, the temperature of the reaction is raised to above 90 degrees C in order to break the hydrogen bonds between strands and produce single-stranded DNA. In the annealing step, the temperature is lowered in order to allow the oligonucleotide to anneal to the target DNA using complementary base pairing. The exact temperature needed for the annealing step is a function of the length and the sequence of the oligonucleotide. In the extension step of the cycle, the annealed oligonucleotide is used as the primer for DNA synthesis. As you should recall, all DNA polymerases synthesize in a 5' to 3' direction, starting from double-stranded DNA. The extension step makes the complementary DNA of the template sequence. The polymerases used for PCR differ from normal DNA polymerases in that they are able to survive the extreme temperatures without being inactivated due to denaturation. Typically, they are cloned from bacteria which live in high-temperature environments, such as hydrothermal vents. In subsequent cycles, the denaturation, annealing, and extension steps are repeated over and over again. At the start of the second cycle, there are now twice as many strands of DNA to act as templates. Half of them end with one of the synthetic oligonucleotides in the reaction mixture. In the second cycle and beyond, the previously synthesized DNA can act as a template as well. However, unlike the original template DNA with overhanging nucleotides of random lengths, the newly synthesized strands have defined ends determined by where the oligonucleotides bind. With the completion of extension in the second cycle, there is now 4 times as much template DNA as we started with. Although the original template is still present, the majority of the DNA in the reaction has been newly synthesized. Strands which used previously synthesized strands as templates are of defined length, because the polymerase ran out of template when it reached the end of the oligonucleotide. The length of these strands is the distance between the 2 sequences in the original target DNA, corresponding to the sequence of the oligonucleotides. As the reaction finishes the third cycle, there are now 8 times as many DNA strands as when the reaction started. Rapidly, the original template is becoming a smaller and smaller percentage of the total template available, and more strands have defined length. The PCR reaction is typically continued through 20 to 50 cycles. Each cycle, the amount of DNA doubles. Quickly, virtually all the DNA in the reaction is of defined length, with the synthetic oligonucleotide on the 5' end. Oligonucleotides put into the reaction don't have to match the template exactly. They can still anneal with a small number of point mutations where the base pairs are not complementary to the template. The oligonucleotides with the mutation are incorporated into the newly synthesized DNA. When they in turn become the templates for synthesis, the resulting duplex DNA has the mutation in both strands. Because of the amplification power of the PCR process, the final reaction contains almost all mutated sequences. This is a very useful method for making changes in the sequence of DNA. If the mutated DNA encodes a protein, then this same technique can be used to make genes, which encode proteins with mutated sequences.